Welcome everyone to Where to with Genomics webinar tonight. My name is Boyd Gudex and I am a technical consultant at BREEDPLAN, and I'll be the facilitator of tonight's session, which will be presented by Catriona Millen of Southern Beef Technology Services. Before we dive in, I would quickly like to deal with some housekeeping. Firstly, if you desire to ask a question and the question box hasn't appeared, please click on the red arrow on the top left-hand side of the webinar screen to get it back. To ask a question, just type it into the question box and press send. And we will be stopping periodically during the webinar to answer any questions that arise. If you are having any audio problems, you can also call in by using your phone by switching your audio from mic and speaker to telephone as shown on the screen, and then dialing in using the number provided. And on that note, I'd like to hand you over to Catriona for the interesting stuff.
Thanks, Boyd. Okay, so as Boyd said, tonight's webinar is called Where to with Genomics. So this is the 6th of the 2016 SBTS and TBTS webinars that we've run this year. So we thought we would do a webinar on genomics because there's been quite a bit of talk about genomics in the beef industry lately. So what we're going to discuss tonight is what genomics is, how it works, the benefits of genomics to cattle producers, the value of performance information when we move to a genomics era, and also we will very briefly cover single-step genomics at the very end. So just before we start, obviously we've got this term genomics. So that's based on the genome. So just to describe to everyone what the genome is, it's all of the genetic material of an organism, so all of that organism's DNA. And this is a picture of Dominette, who was the first bovine to be whole genome sequenced. So our bovine genome is based on Dominette, and it's approximately 3 billion base pairs in size. So the bovine genome is very similar size to the human genome. So just so everyone gets a feel of where we're looking in the organism, we've got our organism here, in this case a cow, and that organism is made up of cells. So within the cell, we've got a nucleus here, and that's where our chromosomes are, and the chromosomes are made up of DNA. So we're looking at the DNA of the animal when we're talking about genomics. And this is just a picture of what DNA looks like. So DNA stands for deoxyribonucleic acid, and it's a double helix structure. It's made up of a sugar phosphate backbone, which we see here in blue, and there's 4 bases. So A and T, which always pair together, and G and C. And that's about all of the biology that I'm going to talk about tonight. So genomic selection uses DNA markers such as SNP, or in this case SNP, to predict phenotype. So that obviously leads to the very valid question of what is a SNP? So a SNP is something we call a single nucleotide polymorphism. And what we've got here is an AG SNP. So if we look at this bull here, we can see his strand of DNA along here. And if we look at this bull, we can see his strand of DNA. Now this DNA is identical apart from where we've got an A in this animal and a G in this animal. So this difference occurs at a nucleotide. It only occurs at one of those nucleotides, and it's polymorphic, so we've got different variants. So that's what we mean when we're talking about a single nucleotide polymorphism. It's a single variant at a single spot in the DNA. Now, it's a little bit more complicated that because cattle, like humans, are diploid organisms. So that means that they've got 2 copies of every chromosome. And so with an AG SNP, for example, we can actually have animals that are homozygous A. So that is, they've got 2 strands of DNA, both with that A allele. We can have animals that are heterozygote. So in this case, this animal is heterozygote AG, and he's got an A at this location and a G in this strand of DNA. And we can also have animals that that are homozygous G in this case. So this animal's got 2 copies of that G allele in his strands of DNA. So there's just the 2 variants usually, but we end up with 3 different genotypes, AA, AG, or GG. So what genomic selection does is it uses these DNA markers, or SNP, to predict the phenotype of animals. And so there's millions of SNP, and they're spread across that bovine genome that we mentioned earlier. And how we genotype them is with this SNP chip technology. So a SNP chip, as we can see here, this is one of the bovine SNP chips, is a chip, and it allows us to genotype a single animal for thousands of SNP markers in one go. Usually when we look at our SNP chips, these SNP markers are spread at fairly regular intervals across the bovine genome, so we're getting fairly good representation of the whole genome when we genotype on these SNP chips. And they also come in different densities. So for example, you might have heard people talk about a 20K or a 50K or a 800K, or alternatively LD, or a low density chip, or a HD, or a high density chip. So this is referring to the density of SNP that we have on those SNP chips. And in the case of a 20K SNP chip, that SNP chip would have 20,000 SNP markers. So 20,000 of those SNP that we just talked about earlier. So using genomic selection, we've used the SNP chip technology to get us those genotypes on those animals. And this has been made possible because the price of genotyping has decreased rapidly in the past decade. So genomics is becoming more likely because it's now a more affordable technology. So genomic selection is widely used in the Australian dairy industry. It was first introduced in Australian Holstein cattle for dairy, and then they've also got genomic selection now occurring for jerseys. In the Australian beef cattle industry, currently only Angus and Brahman are incorporating genomic information into their EBVs, but as we'll touch on later, we're starting to see some of our other breeds prepare to move towards genomics, which is the reason we're doing this talk tonight. So when we have genomic selection, for it to work, we need a reference population. So here we've got some bulls, and in this case, these bulls have phenotypes and they have genotypes. Now, I have just made them bulls, but the reference population doesn't have to just be made up of bulls. It could also be made up of cows. So because these bulls in this case have phenotypes and genotypes, they become our reference population. And they're all of the animals in our particular population that have these phenotypes and genotypes. And we've also got our second group of animals, and these are the animals which don't have phenotypes. And when I say phenotypes, what I'm talking about is the performance recorded traits. So typically these will be young animals which don't have phenotypes. So here we've got our young animals. They don't have phenotypes. So in this case, these young animals won't have been old enough to have been scanned or to have been weighed for, that looks like it's probably a 600-day wait in that photo. But what we can do is we can genotype them. So we're able to take a hair sample and get the SNP genotypes off the SNP chip. So these are our young animals. And how genomics works is the first step is that we can identify relationships between the SNP markers and the phenotypes in the reference population, so those animals that have both performance information and also genotypes. So in this case, we've got 3 bulls. They've got a copy of this A allele from our AG SNP, and we can see that they've also got a high marbling phenotype. And then we've got another 3 bulls that have a copy of the G allele, and they've got a much lower marbling phenotype. So for this particular AG SNP, we can conclude that if an animal has A, it has high marbling, and if an animal has a copy of the G allele, it's got very little marbling. And so when we move to our young animals, we can use those relationships that we've just identified in the reference population to infer the phenotypes of the young animals. So here we've got some young animals. These three have an A allele and these three have a G allele, and we know this because they've been genotyped. But we don't know their phenotype. They're not old enough for us to scan and see what their marbling phenotype is. So we can use that relationship from step 1 to infer the phenotype because we know about the relationship between A and G and the level of marbling. So we can infer that these ones here with the A allele will have high marbling, and the ones here with the with the G allele will have low marbling. Now, this is a simplistic explanation with just one SNP, but we can use the relationships between those thousands and thousands of SNP that we genotype in animals to identify these relationships. So it's not just based on one SNP, it's based on all of the relationships between those SNP genotypes and the trait. So our reference population is therefore critical to the success of genomic selection. And what this shows, along this x-axis, we've got the number of individuals in the reference population, and this is the accuracy of our genomic EBVs in those animals, so young animals, for example, that don't have phenotypes. And the H2 just refers to different heritability. So the heritability of a trait is how much of the variation in that trait is explained by the genetics. So a heritability of 0.01, 10% of the variation that we see in that trait is due to genetics, versus a heritability of 0.8, where 80% of the variation that we see in that trait is due to the genetics. So what you can see is that as the number of individuals in the reference population increases, the accuracy of genomic selection, so how well we can predict the phenotypes of those non-genotyped animals, those young animals, increases. And the other thing to note is that when you've got a lower heritable trait, so a very low heritable trait, for example, fertility traits are usually quite low heritability, so days to calving, for example. We need a lot more records or animals in the reference population to get the same level of accuracy as if we had compared to when we have a highly heritable trait. So this is why we see differences in how well genomic selection works across different traits, because it's much easier to get higher accuracy when the trait's highly heritable. So what this is just showing us is that the accuracy increases, and we're probably looking to get successful genomics that's really helping us in our selection of young animals. We're probably looking around the thousands of records mark for our reference population. So another thing to note is that genomic prediction works best when those young animals are closely related to the reference population. So ideally, when we're setting up our reference populations, we should try and represent all of the variation within the breed and or the population. So to show that, I've got all of these different animals here, and each different colour represents a genetic line within that single breed. So and in this case, it's a fairly small population with not very many different genetic lines. So when we set up our reference population, in this case, if we had these 6 animals that were in the reference population, we've only got 20% of those genetic lines represented in the reference population. So for animals that were closely related to these 6 genetic lines, genomic selection would work quite well. But for example, for animals closely related to this yellow animal or this green animal, genomic selection wouldn't be quite so successful. So what we want to do is try and get a situation like this, where we've got 90% of the genetic lines represented in the reference population. And so that means that a lot more of these animals, so animals related to this yellow animal here, animals related to this green animal here, they've got close relatives in this reference population, and genomic selection will work a lot better for those animals. So, have we got any questions on any of that so far?
No sign of any questions yet.
Okay, so as we're going along, if anyone's got any questions, feel free to pop them in the box, and as they pop up when I stop for questions, Boyd will read them out. So with that, we'll move on to the next section. So there's going to be two main benefits of incorporating genomic information into the BREEDPLAN analysis. So the first of these is that we will be able to generate EBVs for animals which do not have performance data. Now, there's a number of reasons that animals might not have performance data. So the first of these is that animals might be from non-BREEDPLAN herds. So the herd's not performance recording, and we don't have any idea on how that animal is performing relative to its contemporaries. Other reasons that we may not have performance data on those animals yet is that they might not be old enough to be measured for the trait. So, for example, a young animal won't be old enough to be measured for something like 600-day weight, and also probably not old enough to be scanned. Traits which are hard to measure. So, for example, net feed intake, this is typically only measured in our progeny test projects because it's a very expensive, difficult trait to measure. So, a lot of animals won't have their own net feed intake performance data. Traits that are only measurable in one sex is another good example of a reason that an animal may not have performance data. So, for example, mature cow weight, we only measure in the female, so our bulls aren't going to have their own mature cow weight performance information. And another good example of why animals may not have performance data is that some traits can only be measured once the animal is dead. So our obvious example of that is our abattoir carcass traits. So that's the first reason, or the first benefit. The second benefit is that more accurate EBVs can be generated for animals with limited performance information. And a good example of this is a young bull who has his own observations but doesn't yet have progeny. So we've got no observations on the performance of his progeny. So one thing to note is that a genomic test will not improve EBV accuracy across all animals equally. So if we have a look here, we've got two animals. We've got our young animal here, and for this EBV, the young animal has an EBV accuracy of 30%. Whereas if we look at our older bull here, he's got an EBV accuracy for this EBV of 80%. However, if we could do a genomic test, we might see an increase for both of them in the accuracy. So in the case of this young animal, we might see a 20% increase in the EBV accuracy. So with a genomic test, he's jumped from 30% up to 50%. Whereas for this older bull, he's jumped from 80% up to 82%. So we only see a 2% increase in the accuracy of his EBV. Now, this can occur. This occurs for a number of reasons, and it's probably a good time to point out too that this is a hypothetical increase. The actual increase we're going to see is going to be dependent on the individual situation. So the actual improvement in EBV accuracy following a genomic test will vary depending on a number of factors. So as we just saw, the EBV accuracy before the genomic test will influence how much of an increase we see in EBV accuracy once we have a genomic test. So we can think about that like, how much information do we already have? Do we have very little information on that animal? So for example, no information or no own performance, so just pedigree information. And if we've just got pedigree information, adding a genomic test can be like, for example, adding 10 progeny. So we get a big boost in the accuracy of the EBV. Whereas with an older bull, he might have pedigree information, his own performance information, and performance of, say, 200 progeny. So if he's got all of that information and we add the equivalent of 10 extra progeny, we've already got 200 progeny there. So those 10 progeny don't have a huge effect on the accuracy of his EBV. Another thing that will will influence the actual improvement we see in EBV accuracy with a genomic test is the individual trait. So as I mentioned before, lower heritability traits, so traits that don't have so much variation due to genetics, so there'll be a lot of environmental factors influencing the variation that we see in those traits. We need a much bigger reference population to be able to see a big increase in accuracy compared to a highly heritable trait. Another thing that will influence how much improvement in EBV accuracy we see following a genomic test is how closely related the animal being tested is to the reference population. So if it's closely related, we would expect to see a bigger increase in accuracy compared to if it's quite distantly related. And as I've just said, also the size of the reference population is important. So a smaller reference population will see less of an increase in EBV accuracy versus a large reference population. And unfortunately, we won't really be able to give you an idea of what it's likely to be in your breed until we've run a genomics analysis for your breed and seen what happens in the particular breed. So while the improvements in EBV accuracy might vary between animals, we can see that there should be improvements in EBV accuracy. So how does that help you as a breeder? So when we're breeding, what we're doing is trying to improve each generation. And what genomics will do is it will allow us to identify elite bulls and heifers at a young age. And that means that you as breeders will be able to make selection decisions at a younger age than are currently possible. So when we select younger animals, what we do is shorten the generation interval, and that increases the rate of genetic improvement. So genomics will help us to increase the rate of genetic improvement in your animals. So as a practical example of where genomics might be useful for a breeder, here a breeder has implemented an ET program and has produced 10 full sibling bull calves. Now, just to point out that even though they're ET full siblings, they're not identical twins. So ET full siblings don't share 100% of their DNA. They're just like a normal full brother or full sister. They share 50% of their DNA. So we don't expect them to be identical. So in this situation, our breeder's got these 10 full sibling ET calves, and at several months of age, maybe due to drought, the breeder wishes to keep the top 6 bull calves and steer the rest. But the problem is that these animals aren't old enough to have performance information. So how does this breeder decide which ones are the best calves and which ones should be steered? So currently, it's a really difficult decision, as all 10 calves will have mid-parent EBVs and mid-parent selection indexes. So as we can see, without a genomic test, because there's no performance information and their EBVs and selection indexes are based purely on pedigree information, the selection index for all calves is $100. And so the breeder is unable to differentiate these calves based on their genetic potential, so it's pot luck whether or not he picks the top 6 ones or whether he ends up with the bottom 4, as well as 2 somewhere in between. So once genomics is incorporated into the breed plan analysis for this particular breed, then what the breeder will be able to do is to take a hair sample for each calf, pay for a genetic test, and then this information will be incorporated into the breed plan analysis, and EBVs, which include genomic information, will then be produced. So he's now done that, and what the breeder can now do is differentiate between calves using their genetic potential. So the breeder is now able to identify which calves suit their particular breeding program and select which ones are the best to keep and which to steer. So in this case, we've gone for the top 6 with the the 6 with the top selection index. So we can see we've got calf 1 with a selection index of 114 down to calf 8 with a selection index of $98, and these 4 that are getting culled are all the lower selection index animals. So I've used a single selection index for illustrative purposes only, but in reality, the breeder would need to consider their selection indexes, their individual EBVs for traits of importance, and of course our other important traits such as structure. And while I showed you a selection index that had differentiated due to genomic information, we would also expect that the EBVs would differentiate once we include that genomic information. So, have we got any questions?
Yes, we do have a question. We've got a question here of what traits are currently being incorporated in. And currently being tested with the genomic tests. Sorry to clarify.
Yep. So in Brahman, days to calving, and in Angus, I think there's around 20 traits, but I don't know what they are off the top of my head. I think there's some documentation on the Angus website, but if you like, we can find out for you and follow that up tomorrow.
Yep, we've also got another question here of your example of the AG alleles for marbling versus non-marbling. They're asking, surely marbling is controlled by more than one base pair, isn't it?
Yes, it is. That was a very simplistic example of how genomics would work. So marbling is polygenic, so it's controlled by multiple genes. So yes, it wouldn't just be that AG SNP. In that instance, we might see that A is associated with a higher marbling and G is associated with a lower marbling, but I'd expect to see similar effects for thousands of SNP across the genome.
Yep. And one more, who decides what subset of SNPs are going to be measured on a testing chip? The number of SNPs to measure is a relatively small percentage of the entire genome, 3 billion, so therefore presumably the higher density chips are more expensive to use.
Yep. So the companies that produce the SNP chips decide which SNP are going to be measured on that SNP chip. Traditionally, the older SNP chips, so for example, the Illumina 50K, those SNPs were chosen to be spread across the genome quite well, so that the entire genome was well represented, and it wasn't, for example, all clustered on chromosome 1 and other SNPs on other chromosomes not included. They also had to amplify well in the lab, so they had to be reliable SNP that were in good regions of the genome where we weren't going to have problems amplifying them. And that since Dominant was sequenced, there's a number of labs around the world that have done a lot of whole genome sequencing of animals. And each time that's done, they discover more and more SNP. It was starting to plateau, but these early chips, a lot of that SNP discovery hadn't occurred because there hadn't been a whole lot of whole genome sequencing done. So they were also constrained by which SNP had been discovered. Now, I'm going to touch on this briefly in a little bit, but what we expect to see in the future, and it's already starting to happen, is rather than SNP that are randomly distributed over the genome, we're expecting that chips will be developed that have biologically relevant SNP. So that should help with our genomic prediction, because we'll have SNP that are actually doing something rather than a SNP that is nearby another SNP that's doing something. And the other thing about which SNP have been included on the chips is some of the newer chips that we're seeing now actually, and I'll touch on this briefly again, actually include SNP that you can use for parent verification. So some breeds have moved to SNP-based parent verification. And so, for example, on those GeneSeq LD and high density chips, you will see SNP that can do parent verification. A lot of the genetic defects that we see in cattle, so those genetic diseases, for example, a lot of those SNP have been included on the SNP chips, and also some genomic prediction SNP. So, and then the third part of that question was that the high density chips are more expensive to use. Yes, they are. I have been told that the low density chip is around the $50 to $60 mark. I'm not sure if that's inclusive of GST, whereas the high density chip is more around that $110 to $120 chip. But I'm not sure what the costs are for parentage and for individual genetic defect tests, but I expect as breeders are able to do parentage, genomics, and a number of genetic defect tests on a single chip, that the prices will become a lot more attractive as a breeder, because you're not paying so much money for each individual test, you're getting it all rolled into one.
And we've got one more comment here from another participant who says that there is 15 traits are currently available through genomic testing in the Angus breed.
Thank you.
Yep, and that's the last of the questions for now.
Thanks, Boyd. So we'll move on to the next part of the webinar, which I think is going to cover some of what I answered in that last question. So I'm thinking about getting a genomic test done on my animals. What tests are available and what can I expect to get back? So most genomic tests in Australia are handled through either the Animal Genetics Laboratory at the University of Queensland, and AGL is offering the two GeneSeq SNP panels, so the GeneSeq high density panel and the GeneSeq low density panel. And we've also got Zoetis doing genomic tests, and I think they've got an Angus specific test that SNP panel that they offer. And as I just mentioned, I think we'll see this more and more into the future, but there are already opportunities for cattle breeders to do parent verification where the Breed Society allows SNP parent verification, genetic disease status testing, and also genomics on a single SNP chip. And I think the benefit of this is that helps to keep the costs down, in that you're getting a lot of stuff for one initial outlay. So how do I get a genomic test done? Most breed societies have arrangements with service providers, and so the first port of call would be to contact your breed society and see what their arrangements are for getting genomic tests done. But this is the sort of thing that we're expecting. So you will take typically a hair sample from an animal, you'll put it in the post, and it will go to the lab. And once it's at the lab, it will be genotyped. The genotype will be returned to the breed society, and it will be used in the calculation of breeding values. And then the breeder will receive the breeding values back, as you currently do from BREEDPLAN. And so that's the cycle that we're envisaging that we will see. But as I said, most breed societies already have arrangements with their service providers, so they're your best port of call for getting the forms and things like that, and costings as well. So what you won't get back is the actual genotypes. These will be coded as very long strings of 0, 1s, and 2s. So, for example, if you do a 50,000 SNP chip test, you will get, well, you won't get, but there would be 50,000 0, 1s, and 2s that will come back to the Breed Society. So, as you can see here, we've got animal ID 1 with this string of numbers. The same for animal 2, animal 3, animal 4, and animal 5. So, to most people, this doesn't mean much. So you won't see these because you couldn't do anything with them anyway, and they are very large files. And for someone, for example, with a herd of 200 animals that's recorded, that's genotype 200 animals, you're going to get a lot of 0, 1s, and 2s back in large files that you couldn't do anything with, and they'll take a lot of space up on your computer. What you will get back is EBVs. And so these will be calculated not just from pedigree information, the individual's performance information and the progeny performance information as they are now, but also from genomic information. So what genomic tests will be available in the future? I expect, I'd like to have a crystal ball so I could tell you what will happen, but what I expect will happen is that we'll see SNP chips continue to evolve, and there will be new SNP chip panels come onto the market in the future. And as I mentioned earlier, we expect there will be a focus on biologically important SNP markers. So I think there's one due to come out in the US later this year, and that's focused on some biologically important SNP markers for fertility traits. And I would like to think that they will be cheaper, but I think we're going to have to wait and see on that one. So another point that I'd like to cover tonight is performance recording. So I'm going to go ahead and do some genomic tests on my animals. I'll get EBVs that include genomic information once my breed moves to genomics. So now I can stop performance recording, right? Wrong. So this is the short answer, no. And Professor Mike Coffey from the Roslin Institute in the UK, I think this has been paraphrased a lot since he said this, but in the era of genomics, phenotype is king. And as we said earlier, that phenotype is our performance recording. So earlier we were talking about the reference population being vital to the success of genomics, and that genomic prediction will work best when those young animals are closely related to the reference population. So what this means is that over time, that reference population will need to be continuously updated. And so animals with both genotypes and phenotypes will be needed. And as we move to genomics, I'm going to talk about single step a little bit later, but as we move to single step genomics, that reference population will be made up of any animal that has a genotype and a phenotype. So for example, if you take a genotype on your animal and you collect phenotypes on that animal will be included in the reference population. And that's a good thing because that means that your animals will be closely related to the reference population because you've got some animals that, of your own lines that are in that reference population. So that's the first reason that performance information is really important and that we don't want people to stop performance recording. The other thing is that when individual breeds move to genomics, EBVs will be calculated from the pedigree information, from the performance information on an individual animal, and from the performance information on the progeny, but also from that genomic information. And in this fashion, genomics can be thought of as just another source of information. So genomics will give us the opportunity to have more accurate EBVs earlier in an animal's life, but it's highly unlikely that these are going to be high accuracy EBVs of an accuracy of 90% or more. So just as you wouldn't stop collecting performance information on an animal just because it has 10 progeny with performance records, we recommend that you don't stop performance recording once you've got a genomic test done. So even once you've got that genomic test done, other performance information will still be valuable for the calculation of your EBVs. So do we have any more questions?
Yes, we do. We've got a question here. Are structural traits, e.g. cows with 2 weak quarters, undershot jaws, etc., created by genetic defects and parents' coat colours not normal within breeds be identified, or is it just traits measured by EBVs, or for EBVs, sorry?
For genomics?
Yeah.
So genomics will contribute to the EBVs that you currently have. So I wouldn't expect coat colour or structure through weak quarters to be traits that we identify through genomics because we'll just be using the genomic information to calculate EBVs. that said, I'm not sure about the structural one, but some of these SNP chip tests may include coat colour tests. And if that was the case, if you did a SNP chip test or got a SNP genotype, you may also be able to select to do a coat colour test at the same time.
I'll just add something to that as well. There's certainly things like some of the genetic defects we are now aware of and have tests for are often included on these chips. So in a lot of ways, most of them are structural traits that are included, but your specific ones, not exact.
Thanks, Boyd. Okay, so this is the last section before we finish up. So as I said earlier in the talk, Australian beef cattle, currently only Angus and Brahman are incorporating genomic information into their EBVs, but we've got a number of Australian beef breeds who are currently moving towards introducing genomics in the foreseeable future. And as we head towards the future, the genomics methodology that we're using will continue to be improved. So with the breeds that are currently running genomics, we're using what's called a blending method. So the genomics information is blended with the EBV. Now there's a number of limitations with that method, and one of these, which I think is the most important for breeders, is that the genomic information only influences the EBV for the animal which has been genotyped, because the traditional EBV is blended with the genomics information. In the near future, we're expecting that the single step method will be available, and in single step, all of that information is used together in the one calculation. So this will mean that genomic information will flow through the pedigrees and therefore can influence the EBVs, not just of the animal that's been genotyped, but also of related animals. And it's worth noting that with the majority of breeds who don't currently do genomics, I expect that the majority of those will move straight to a single step methodology once they implement genomics. So what this means for you guys as breeders is that basically the improvements in methodology will contribute to more accurate EBVs. And that's a good thing, because once we have more accurate EBVs, you can be more confident in your selection decisions, particularly if we can get those more accurate EBVs in younger animals. So in conclusion, genomics relies on a well-designed reference population. So these are our animals with genotypes and phenotypes, or performance information. And there will be two main benefits of genomics for breeders. EBVs can be generated for animals which do not have performance data, and as we mentioned earlier, that can be for a variety of reasons, such as they're not in a BREEDPLAN recording herd, they're too young to have that trait recorded, the trait can only be recorded in one sex, the trait can only be recorded once the animal is dead. And the second benefit is that we'll be able to generate more accurate EBVs that can be for animals with little performance information. So a classic example of that is those young animals that have their own performance but don't yet have progeny. And so genomics will therefore allow breeders to make more informed selection decisions on young animals, and this in turn allows the generation interval to be shortened, and that in turn allows for an increased rate of genetic improvement. So as individual breeds move to genomics, EBVs will be calculated using not just pedigree information, individual performance information and progeny performance information as they are currently, but also include genomic information. And as we've just talked about, that performance information will continue to be critical in the genomics era. So I think that, I think of genomics as just another source of information. It's like adding some progeny records for an animal. It's not the only source of information we want. It helps to get us more accurate EBVs, but we need to keep collecting as much data as we can, even once we move to genomics. And lastly, you won't be able to see your genotypes because they're going to be in strings of 0, 1s and 2s, which don't make sense to anyone, and also because they're going to be very, very big and require a lot of storage area. But what you will see is that this information will be incorporated into the calculation of your EBVs. So if anyone's got any further information, you can contact the relevant SBTS or TBTS technical officer. So for Limousin and Simmental, that's Alex McDonald. For Blonde, Charolais, Devon, Hereford, Murray Grey, Red Angus, Red Poll, Solaires, Shorthorn and Speckle Park, you can contact me. For the tropical breeds, so Brahman, Brangus, Droughtmaster, Santa, Senapole and Simbra, you can contact Paul Williams, and Carl Tesling is the SBTS Technical Officer for Wagyu. And just before we finish, Rob Banks, the Director of the Animal Genetics and Breeding Unit at the University of New England, did contribute several slides, so I'd like to thank him for letting me steal his slides. And if there's any more questions, we'll pause now for questions.
We have a question here of why not pool all Bos Taurus animals regardless of breed for a greater pool of animals for research?
That is a good question. So unfortunately, they did try this in dairy, and it doesn't work very well. And I think I'd have to get quite in depth into biology as to why that doesn't work, but I'm going to give it a try. So generally, the SNP on the SNP chips aren't the causal variants. And so in some breeds, say for example, we've got an AG allele, which is our SNP, and then we've got our quantitative trait nucleotide, which is big Q and little q. In some breeds, the A will be associated with big Q, and in other breeds, the T will be associated with big Q. So we call this phasing, and this means that just because the A allele works well, for example, in Hereford to predict a certain trait, it might be the G allele in Angus that predicts that trait. So we have problems with differences in breed, and we also have variation that's found in some breeds and not in other breeds. So while it sounds great in theory, unfortunately it doesn't work very well, which is a shame because, as you've pointed out, if we could pull all of those animals together, we'd have a much bigger reference population. But at this stage, we can't do it. That doesn't mean we won't be able to do it in the future, but it is difficult, and at this stage we just don't have the technology to deal with it. And the other problem that we can potentially have is if breeds aren't represented equally in that reference population of multiple breeds, so say it's 90% Angus and the other 10% is made up of those smaller breeds, we could see Angus having a much greater influence on the phenotype predictions because they're a greater share of that reference population. So I hope that answers your question, and I hopefully haven't confused you by talking about phases, but if I have, let me know and I'll try and explain it a little bit better.
And we have another question of how close is beef to being able to completely identify parentage using a SNP test? Dairy is doing this.
As far as I'm aware, there are some beef breeds or breed societies that have implemented SNP parentage testing. I'm not sure which ones have implemented SNP tests and which ones are still using microsats. One of the problems is that transition from microsatellites to SNP, where you have some animals that have only microsatellites and you can't convert a microsatellite to a SNP. So that means generally that those ones that have microsatellites need to be retested using a SNP test. But as far as I'm aware, it is possible.
I can confirm that completely. I've actually been involved in developing a test in another livestock species, and where we had to go through doing that. And the biggest problem is resistance of breeders who go, I've already got a marker profile for my cows, but it's in the old format, the microsatellites. Why do I have to redo it for SNP? And unfortunately, they're completely different technologies. So you have to not only pay for the new test on your new calves, you've got to pay for it on the parents that you're matching them to as well. So that is a big factor in it. We have one more question just come in. How do you see the future structure of the breeder population changing to prevent freeloaders, aka those who use only genomics, taking advantage of the hard-working breeders who continue to do phenotypes and genomics? Right, well that appears to be the end of our questions for now, so we'll just swap and finish some housekeeping, and please feel free to continue sending in questions before we sign off, and if we see them, we'll get them to them for you. There will also be some feedback on tonight's webinar. There's a poll that will be opened up upon completion for you to just answer. I think it's 4 questions, so it's nice and quick. And I'd like to reiterate, please feel free to contact any of the SBTS or TBTS staff with any more questions you have. And these webinars will also be available on YouTube for those of you who want to look at them again. So I'd like to thank you all for listening, and I'll just double check to see if there is any more questions. There doesn't appear to be, so I think that is it for everyone. Thank you very much for listening.
Thanks everyone. We'll finish the webinar there.